Hello, everyone, and welcome to this Genetic Engineering and Biotechnology News webinar. It's entitled The ABCs of Glycoproteins, How to Combine Analytics, Biology, and Chemistry to Improve Your Understanding of Glycosylation. With over half of all known proteins requiring glycosylation, it's easy to see why this is one of the most important and critical post-translational modifications. It's also extremely complex and absolutely necessary for proteins to function properly. To help you better understand all aspects of the glycosylation process and to offer some suggestions on how you might carry out your own glycosylation projects more efficiently and more effectively, we've put together today's webinar. Our two panelists are our glycosylation experts, so let's meet them. Dr. Pauline Rudd is Research Professor in Glycosylation at the National Institute for Bioprocessing Research and Training in Dublin, Ireland. Dr. Rudd is going to talk about multidimensional glycosylation analysis. Dr. Ying-Ching Yu is Principal Scientist at Waters Corporation. She'll be discussing biotherapeutic protein glycosylation analysis using UPLC QTOF mass spec. I'm John Sterling, Editor-in-Chief of GEN, and I'm going to serve as the moderator. Please feel free to enlarge the slide images or download the complete presentations. At any time during the webinar, you can send in a question for our panelists. Type your question into the Ask a Question box on the lower left of your console and then hit Submit. The panel will try to answer as many as possible during the question and answer segment that takes place after all the presentations have been made. So if everyone's ready, let's get going. Dr. Rudd will be our lead-off presenter. Pauline?
Thank you, John. So I'm very happy to be introducing you to my favorite subject, glycobiology, and in particular the analysis of glycosylation, which is a challenge for everyone, including my group here at the NIBIT Glycobiology Group within the National Institute for Bioprocessing Research and Training. So just to fill you in as to who we are, the NIBIT is a private institute which is based on an innovative partnership originally between 4 academic institutions, and it was funded by Ireland to the tune of 57 million euros. And the idea of NIBIT is that we will conduct world-class research in key areas of bioprocessing, and that we will have a facility in order to be able to train highly skilled personnel for bioprocessing and also to provide flexible multi-purpose research and training facilities. And so our part in this enterprise is to provide bioanalytics training and in particular glycan analysis. So just to fill you in with a bit of background, so everyone knows about DNA. It's made up of 4 bases. Although it's a huge molecule, 3 kilometers in each cell, and each adult has enough DNA for 70 trips from the Earth to the Sun and back. But nevertheless, this is a linear molecule and it's relatively straightforward to sequence. Likewise, amino acids, which the DNA codes for, these are also linear structures. They're made up of polypeptides which fold into proteins. And then the magic ingredient are the post-translational modifications, in particular the N-linked glycans and the O-linked glycans, which are attached co-translationally. And here you can see a picture of erythropoietin. You see how huge the orange sugars are, both the N-links, the 3 N-links and the small O-link. So why are we so interested in glycoproteins? Well, therapeutic glycoproteins are secreted recombinant proteins that are intended to interact with the host. And of ones on the market at the moment, 127 biological therapeutics are glycoproteins. And you see here they span everything from blood proteins to growth factors to monoclonal antibodies. So it's very important to understand the processes which result in this glycosylation. So the most well understood is the attachment of N-linked glycans to the nitrogen in the side chain of asparagines. And this event happens in the endoplasmic reticulum, and the asparagine has to be in a little triplet, asparagine, some other amino acid, and then either serine or threonine. O-linked glycans are a bit different because they're attached to the folded protein after it enters into the Golgi apparatus. And here you can see on the page a typical glycoprotein that actually is a therapeutic tissue plasminogen activator. And this illustrates one of the first complexities of glycan analysis. It has two different forms, type 1, in which 3 glycosylation sites are filled, and type 2, in which only 2 of the sites are occupied. And this is known as macroheterogeneity. The first thing to know about your protein is how many of these potential glycosylation sites are actually occupied. And the second molecule there is IgG. This is a typical monoclonal antibody type structure. And you can see in the CH2 domains, you can see the sugars, one on each heavy chain, buried in the middle of those domains. So how do the sugars actually come to be processed? Well, first of all, when this oligosaccharide transferase enzyme sees this little triplet of amino acids, it recognizes that this is an opportunity to attach a block of pre-formed glycans onto the nitrogen on the asparagine side chain. Once it's done that, a number of enzymes cleave back this preformed block until the protein's properly folded and the whole neoglycoprotein moves into the Golgi. There are then other enzymes that you see in the bottom of the figure which are there to build up these arms of the glycans to give more complex type sugars. So gluconac transferase 2, fucosar transferase 8, acylar transferases, they all process what are known as complex type glycans. And here you can see a biantennary structure, but you can also have tri and tetra and extended structures. So these sugars can be very large. And what makes things more complex is that some of these enzyme reactions don't always go to completion. So different copies of the glycoprotein going through this pathway will end up with what's known as microheterogeneity. In other words, at each glycosylation site, there'll be a bunch of different sugars, and this is the result of incomplete exposure to the enzymes which process the sugar chains. So if you look at erythropoietin in a bit more detail, this is a secreted glycoprotein. Glycosylation is cell, protein, and site-specific. So in other words, if you express the same glycoprotein in different cells, you get different glycosylation. If you express different proteins in the same cell, you get different glycosylations. And if you look at each site here on erythropoietin, you can see that even the type of glycosylation at each site is different because the three-dimensional structure modulates the processing of the glycans at each site. And glycans can be also affected by the environment. So in a bioprocessing situation, they can be affected by the conditions within the reactor. The role of the sugars in the case of EPO is that they extend the half-life. If you have the wrong sugars there, in other words, if they're not tetraantennary and they're not tetrasylated, then the half-life is reduced from 3 hours down to 3 minutes, where it's barely efficacious. The other role of the sugars is that they stabilize and protect the protein from protease activity, and in some cases they can modulate the activity of the protein. And if you're expressing your protein in non-human cells, then you may introduce antigenic epitopes. So sugars are quite complex because of this macro and micro heterogeneity. They're also hard to analyze in their own right because two sugars can be linked together in several different ways. So they can be linked at the 2, 3, 4, or 6 positions. And the way in which they're linked together can either be above the plane of the ring or below the plane of the ring. So as well as defining the particular monosaccharides and the carbon numbers which are linked together, you also need to define the anomoricity at each linkage. And to make it even more of a problem, glycans are branched because several residues can be linked to a single monosaccharide. So they're really a different class when it comes to comparing the analysis of glycans with DNA and proteins. It's also quite important to use a symbolic representation that works well for you on the bench. And this is the one we use. It includes monosaccharide sequence and linkage information, and it also works easily in black and white, so that's a help if you're working with a pencil on the bench trying to define structures. You can see with the symbols, all the hexoses are actually open symbols, and the hexnacks are filled in symbols, and this helps you to easily add up the composition for mass spectrometry. The linkage position is defined by the angle in which two monosaccharides are linked together, and you can see the difference between an alpha-1 3-link fucose you find in plants and an alpha-1 6-link fucose that you see in mammalian cells. And another good example of this nomenclature is that if you look at the difference between sial-Lewis A and sial-Lewis X, you can see they're the same 4 monosaccharides in each epitope, but they're linked together in different ways. So although they have the same mass spec composition, they are in fact 2 different structures. So there are many reasons why you might want to do glycan analysis, and so it's very important to understand your question before you decide which analytical techniques to use, because there are very many approaches to glycoanalytics, and they all work, they all give you some information, and you need to understand what information you want before you embark on using a particular set of technologies. So you may, for example, be in basic research. You might want to be able to understand the role of the sugars in the function of the protein, in which case you'll probably want quite detailed glycan analysis. If you're doing cell line selection, then you're probably looking for high-producing cell lines with the appropriate glycosylation that you've already defined. And if you're looking for safety and efficacy, then again you might be highlighting some particular epitope. For example, this is an alpha-gal linked sugar which is antigenic in humans. When you come to production and scale up, then all kinds of things can affect this: the media composition, the temperature, the pH, patches of hypoxia in the bioprocessor, and so on. So you'll need to be understanding the relationship between media components and your glycan signature. When you come to process analytical technologies, then you need to have outline monitoring. You need to be able to identify changes so that you can troubleshoot if the glycans are clearly not being processed properly. And then when you come to going to the regulators or releasing batches, you need very careful product analysis that defines quantitatively the critical features of the molecule. And you could be working in product stability when you need to check the integrity of the glycans during storage. And then coming full circle, you need eventually to test for function to understand how your antibody is interacting with a range of efficacy receptors, and this is getting close to basic research again. So these are many different reasons for doing glycan analysis, and there are really 5 stages. First of all, sample prep. Then you need to release the glycans. Then you need to label them if you're going into LC technologies. You need to be able to separate them, and then you need some means of doing data interpretation. So this is a paper that we wrote where it might be a good place to start. We tried to explain how you would systematically approach protein glycosylation. So this is basically the system we use. We immobilize our glycoprotein either in a gel block or now mostly on solid supports in a 96-well plate. We reduce and alkylate. We release the N-link glycans. We use a chemical method to release O-link glycans. We then can take this pool of released glycans and we can label it with APTS to do capillary electrophoresis. We can have it unlabeled if we want to do mass spectrometry. We can use reverse phase HPLC, or we can put a 2AB label on, and we can then do weak anion exchange chromatography, or we can go into lipophilic separations on hillock columns, and then we can use our databases in order to interrogate the pool of released sugars that we have from the sample, and then we can confirm those preliminary structures by using a series of exoglycosidase digestions. So I'll just go through that in a little bit more detail. So the next slide shows you our optimized automated glycan release and labeling robot. And we have this so that we then can go into our analytical separation, and we have computer-assisted data analysis that fits with this. And this means that pretty much everything is automated. It works on the principle, the separations technology works on the principle that we have either a TSK amide 80 column or more recently the BEH columns developed by Waters. And we run a dextran calibration ladder that you can see running from 3 to 10 or more glucose units. We then separate our pool of sugars from our sample. This is released glycans from IgG. And we then run those out on the same column. And that enables us to use the software to assign glucose unit values to each of these peaks. So you can see a peak here at about 110 minutes. And by matching it with the ladder above, it has a glucose unit value of about 8.4 glucose units. And these columns and the BEH columns are very high resolution, the BEH even better than this. But even this column will separate arm-specific isomers. So it makes these columns very useful for determining differences in isomers that have the same mass but actually different structures. So the enzyme digests, they work like this. We have a series of enzymes that you can see here, sialidases, fucosidases, and so on. And they're very specific for the terminal monosaccharide and also the way in which that monosaccharide is linked to the next sugar. So if you use arthrobacter-sialidase and NAN1, you can use these to chop off the sialic acids that are attached to your protein. And you can see they have different specificities. So by using both of them, you can determine whether you have a 2,3 or a 2,6 linked sialic acid. And you can then continue with the fucosidases, which will distinguish between 1,6, 1,2, and 1,3 linkages, and the galactosidases, and finally the hexosaminidases. And the idea is to use these enzymes in panels so that in the end you just arrive at the trimanosyl core, which is common to all N-glycans. So we have a database which is open access, which allows you to go online and find out the glucose unit value for maybe 400 different N-linked glycans. It will also tell you the effect of the enzymes on those particular structures. And it will give you the mass spec, it gives you capillary electrophoresis values. There's all sorts of interesting information there. And attached to the Waters Unify software, there's a Waters UPLC collection, which is a collection of molecules which are of some interest to pharmaceutical companies. And if you go into this page, you'll be able to find out the glycans that we found associated with Herceptin, for example. So it's an aid, really, if you're just beginning to work on these kinds of molecules. Here's an example of Herceptin, and this was some work done by Mark Hilliard, where he took the top line, which is the undigested released N-glycans, and then treated with a sialidase. And you can see where any peak moves, then that tells you that that peak contained sialic acid, which has been removed by this enzyme. And you can then do the same with the fucosidase, with the galactosidase, and finally with the hexosaminidase. And you can see his arrows are going the other way, and that's because sometimes when you're interpreting it, it's actually easier to go from the bottom to the top. But if you study that profile, I think you should be able to follow how this method works. Now, the reason that everyone's so interested in terminal residues on N-glycans on IgG is because most of them now are known to have some function in modulating the effector function of the IgG molecule. So if you have IgG sugars which are lacking fucose, you can encourage ADCC activity. It's decreased by 50% if you put a fucose in the core. So if you want ADCC, you're very keen to avoid having core fucose. There's now evidence to support the idea that these molecules have anti-inflammatory activity. If you have terminal galactose, then these allow your molecule to bind to the FcRn receptor and also increase C1q binding, open up the complement pathway. If you have IgG sugars terminating in N-acetyl glucosamine, then these are ligands for the mannose binding protein and potentially can activate the complement pathway. And down at the bottom again, you can see the alpha-gal antigenic residue. So just to look at one thing which is particularly interesting, and this is the way that the glycosylation of IgG affects Fc gamma R3A interactions. So sideways is the IgG molecule with a circle around the sugars in the CH2 domain. And then in blue is the Fc gamma R3 receptor. And you can see how these two surfaces have to come together in order for IgG to interact with the receptor and to cause ADCC. Now, some very elegant work by Sonderman and Ferrara has shown that the lack of core fucose in these sugars just increases the affinity of this junction because it unblocks some steric hindrance in the top of the CH2 domain. So if you like elegant crystallography, then have a look at this Sonderman paper. So you might be asking by now, well, what are the influences that affect the composition of glycomes and glycoforms? Because clearly it's very important to understand that because the glycans, particularly on IgG and EPO, do affect their function. So these range all the way from genes, single nucleotide polymorphisms, epigenetic factors such as demethylation. The next level, they can be changed by expression levels or activities of processing enzymes, and there are about 600 other proteins involved in protein glycosylation. So this is a very big effort on the part of the cell, and if any of these factors are messed around too much, then they will affect the glycan structures. Also, the levels of the monosaccharide nucleotide donors that bring the new monosaccharide to be added on to the developing sugar, metabolites and lipids in the medium, organization of cellular organelles is very important. If they become mixed up, as they often do in cancer, then you get huge changes in glycosylation. There are many things that activate cell signaling pathways like cytokines, drugs, and oncogenes. And, of course, the environment in bioprocessing and in people, the effects of disease also have readouts in terms of glycosylation. So now more and more in patients, we want to be able to link these different omics together, but also I think when people are dealing with cells more and more, they'll be interested in being able to link these different pathways to make sense of the way in which their product is being made in the cell. Just a few factors in the medium and that can change glycosylation during production. It's well established that ammonia concentration is important. The growth rate of cells is important. The amount of dissolved CO2, the media composition, the times at which the batches are being fed, patches of hypoxia, and then anything that you deliberately do to change glycosylation, like deleting a glycan enzyme or inhibiting its activity, and the number of days of the culture. These are all things that are well known to affect glycosylation. This slide shows you the difference between different erythropoietins that are on the market. You can see here just by using a simple HPLC analysis with no other information, you can see that these different human and CHO cells have different glycosylation signatures, and sometimes that's the only analysis you need to give you the kind of information that you want, which is how different are these EPOs from each other. You may also want to use a very simple technique to identify an antigenic epitope. Here at 306 mass M over Z values is the diagnostic ion for this potential antigenic epitope, N-glycosylated acid. Sometimes, particularly in EPO, you want to be able to characterize the relative proportions of different charge structures. And here on the left, you can see a simple 1D weak anion exchange analysis of the release glycans, which will give you the monosylated pool in blue, the di, tri, and tetrasylated pools in yellow. If you want more detail, you can take each of those individual pools and run them on a HILIC or UPLC system, and this rather simplifies the glycoanalytics. If you take the tetrasylated pool, which is in orange down at the bottom, you can use the enzyme array digestions that I just described to go into a third dimension of glycan analysis. And here you can see, if you know that this tallest peak in the top profile is this large antennary tetrasylated structure, you can work your way down through these different enzyme digests, and you can see how we can demonstrate that that is in fact the right assignment. Then if you need to go into more detail, which you sometimes would if you were deep into research, you can use negative ion CID spectra in order to determine which arm of a tetrasylated structure the sialic acids are attached to, for example. And then you could go even deeper into looking at the number of OSO acetyl groups which are substituted into the sialic acids. And this is a multi-mass spec profile. So I'm giving you these examples of how many different technologies you can use and how deeply you can get into the glycoanalytics. So before you start getting as detailed as this, you need to understand what exactly it is that you need to know. Here is a glycan analysis of a molecule which is very much in everybody's minds at the moment. It's etanercept. It's a TNF-alpha receptor, and it's fused through a mucin-like domain to an FC region of IgG. In this mucin-like domain, there are at least 32 glycans per molecule, and they mostly are these core-1 type structures. Here is a combination of technologies which is now becoming very well used and very useful. Here in the top profile you can see a separation of the released N-glycans from etanercept on a Waters BEH column. And then in the bottom profiles you can see that one of these peaks has been separated, and it's been analyzed on the mass spectrometer in one dimension, but then there's fragmentation has happened in MS to the 2. And this is all done online. And the information in the new unified programs, which are coming out from Waters very shortly, will enable you to take LC information and assignments from the databases from the LC profile, and then the mass spec will then confirm these online. And just to show you how complex O-glycosylation can be, here is what I would call 6 dimensions of glycan analysis. This is the chicken mucins from the large intestine, and each of these glycans released from the mucins was analyzed independently using mass spec to the nth. So here you can see one particular peak that was then identified using mass spectrometry, and it turned out to have to be both sulfated and sialated. And this is really state-of-the-art glycoanalytics. And it enables us to see that the chicken intestine has very many more sulfated structures than the human intestine. And this explains in part why Campylobacter is a commensal in chicken, but actually very toxic to humans. So if you really get into glycan analysis, then this is where the cutting edge is, and this is where the real challenge is to the analyst. And so I'd just like to end by thanking the key people in the glycobiology lab in NIBUT who provided the data for most of the work that I've shown you. Thank you very much for listening, and I'm now going to hand you back to John.
Pauline, thank you for that very detailed overview of glycosylation analysis. You've covered a number of important topics, some of which will also be addressed by our next speaker. So thank you so much. If you're just now joining our webinar, welcome. As I mentioned earlier, we'll be conducting a Q&A segment following the panelists' presentations. Please type your question for any or all of our panelists into the Ask a Question box on the lower left of your console, and then hit Submit. Our second panelist, Dr. Ying-Ching Yu, will now begin her presentation. Ying-Ching?
Thank you, John. My presentation is titled Biotherapeutic Protein Glycosylation Analysis using UPLC QTOF mass spectrometry. So this first slide shows why glycosylation is a key critical quality attribute. About 40% of all approved therapeutic proteins and 8 of the top 10 selling biologics of 2010 contain end-linked glycans. Glycosylation is the key critical quality attribute for biotherapeutics that are glycosylated. It has profound impact on the drug safety and efficacy. The International Conference on Harmonization Guideline Q6B requires the analysis of carbohydrate content, structure profile, and the characterization of the glycosylation sites within the polypeptide chain. In this presentation, I will present the current and the new analytical solution from Waters Corporation for glycan analysis at intact protein, release glycan, and the glycopeptide level that addresses the ICH guideline. So this slide shows a UPLC and MS-based analytical workflows for comprehensive glycosylation analysis. We typically start with glycoprofiling at the intact protein using fast or rapid reverse phase LC-MS. We then perform helic LC separation of the enzymatically released N glycans that are labeled with the fluorescent tag. For this workflow, a fluorescent detector is used to obtain fluorescent chromatogram for profiling purpose. In Professor Pauline's presentation, she gave a very comprehensive description about how to assign to AB labeled glycan structure using a combination of exoglycosidase digestion and a glucose unit retention time assignment, done using either UPLC or HPLC with fluorescent detector. Now, the UPLC fluorescent detector can directly interface with a mass spectrometry, such as a QTOF mass spec, to get MS chromatogram for accurate mass measurement. The glycopeptide, the last workflow shown in this slide, the glycopeptide generated from protein proteolysis are subject to both collision-induced fermentation and electron transfer dissociation to obtain glycosylation site and the site heterogeneity information. So this is typical workflows from Waters. And here is routine LC-MS analysis of intact protein that is typically done using a QED UPLC H class with UV detector and a Xevo G2S QTOF mass spec. The column chemistry is BHC4 reverse phase column. MassLink software is used for data acquisition, and the BioPharmaLinks version 1.3 is used for post-run data processing using MaxN1 deconvolution algorithm, which converts multiple charged ions to charge zero neutral mass. Here's in the bottom is an example of how the raw spectrum with charge envelope and the deconvoluted spectrum looks like. So the deconvoluted mass shows a glycan distribution within in this case, is a monoclonal antibody. So this slide shows an example of comparing 3 batches of monoclonal antibody from trastuzumab. Using the intact protein workflow, this figure shows after BioPharmaLinks data processing, and these are the deconvoluted spectrum from these 3 batches of trastuzumab sample. And you can clearly see in batch 2, there is more high mannose type of glycans that's not as intense in batch 1 and batch 3. We also perform partial reduction of the monoclonal antibody to obtain light chain and heavy chain prior to LC-MS data acquisition. The goal to do this sample manipulation is to reduce the size of the monoclonal antibody to get better MS spectrum In addition, we're now only looking at one glycosylation from the heavy chain instead of a combination of two. Here's what a chromatogram and spectrum looks like for reduced IgG1 antibody. You have light chain chromatogram and heavy chain chromatogram. And if you sum the heavy chain chromatogram, you get the charge envelope. And here is a zoomed-in spectrum of a plus 34 charge states, and it shows the individual glycoform from this heavy chain. And the maximum deconvolution from biopharmaceutics shows same differences we saw from the intact protein is that batch 2 has more high mannose. The man5 peak stands out a lot stronger than batch 1 and 3. So this gives some indication the change from batch 1 and 3 to 2, it could cause by some changing in cell culture conditions. Now, I switch gear a little bit, talk about fluorescence label glycan analysis. So we talked about intact protein glycoprofiling. Now I will work you through how to do FLR glycan profiling using Waters' analytical system. So we start with sample prep. The fluorescent labeling is done using 2AB, just same as Professor Pauling's method. And we offer a GlycoWork sample preparation kit from Waters, and there's a web link for anybody that's interested. This kit helps scientists to do free glycan extraction and 2AB labeling and removal of excess dye and prior to UPLC helix separation. So after the sample prep, the sample were separated on the UPLC system with the BH glycan column chemistry and the UPLC system's H-class bio. And you can see this can be run on the Empower system. I think we have a lot of scientists out there using the Empower system to do glycan profiling, and it could be QA/QC purpose or it could be confirmation purpose. And one nice thing is that you can use Professor Pauling Roth's workflow to do GU value assignment in this workflow. And then here, just comparing my released glycan data with intact protein data now. This is the 3 batch of transduce map. And now we're looking at the released glycan profiling. That's the overlay of 3 FLR chromatogram. And again, just to show you here is that I do see high abundance of mannose 5 and man 6 and 7 and 8 in batch number 2 versus batch number 1 and 3. That confirms the intact protein data. And here we did a relative quantitation of looking at the fluorescent chromatogram profile and the heavy chain intact protein deconvoluted data, and we also did a half FC deconvolution. I did not discuss the experimental detail, but we did compare all 3 methods and looking at the common glycan distribution, and the statistical analysis shown in this chart shows that all 3 methods give very, very close and consistent glycan quantitation, relative quantitation value. Now, here I wanted just to point out why using UPLC, what's the advantage doing UPLC with the BH glycan column. We have looked at, compared HPLC separation with HPLC amide column versus UPLC with UPLC BH glycan column. And we scaled the flow rate according to the column dimension. And this is an example to show that the 2AB labeled human IgG glycans, you can get really good separation. You see most peaks are baseline resolved using UPLC technology. Those peaks are, for example, 5, 6, and 7. They're not baseline resolved in HPLC system. And here's additional benefit coming from you adding interface FLR detector to mass spectrometry. So the scientists can acquire both FLR and MS chromatogram all in one single injection. Now, not only you can do relative quantitation with FLR trace or do GU time assignment, now you can add the benefit at getting accurate mass measurement for each peak component. So here just to say, I find it's very helpful when I do this type of analysis that I have fluorescent chromatogram, it only tells me that, those are the glycans that are fluorescent labeled, and I can go down to the MS trace and sum the scan underneath the peak that lines up between the FLR and MS trace and get the accurate mass measurement. There is some information on this particular application note. If you're interested, you can go to Waters' website, find some detail. Now I switch gear a little bit more. Now we talk about intact protein and release glycan. I will touch a little bit on the glycosylation site characterization. So everybody knows CID for glycopeptide tends to generate neutral loss ions versus ETD will keep those glycosylation modification inert with the peptide and just giving the peptide bond cleavage. And here's an example to show. This is one of O-linked glycopeptide from EPO. Underneath the CID, if I do CID on this peptide, I saw sequential neutral loss from the O glycans that attach to the serine, and also these neutral losses counterparts as the ions in the low mass range. Those are called oxonium ions. And under the ETD, and we can see C and Z ions, and they give pretty much almost a de novo sequence along the peptide backbone, and also from C9 and C10 ion mass shift, you can see it's coming from serine plus a glycanac, a hexose, and a salicylic acid. So that tells you where exactly the glycosylation site is located. And here's an example of ETD spectrum of N-linked glycopeptide. This is actually from trastuzumab. We have application note for any person that's interested in detailed information. So this shows that ETD of N-glycopeptide is possible, and you can select precursor ion N+4+ ion, and after the ETD fragmentation, you'll see nice C and Z ion distribution. And there's no neutral loss from this G2F glycan moiety, which is very good. And here's an example that the scientist is possible to do a highly glycosylated peptide fragmentation using ETD on our Synapse G2S HDMS system. So this is a peptide from Etanercept. It has 7 glycosylations. After these peptides being subject to ETD fragmentation, we get very nice C and Z ion fragmentation, ion distribution. And it's not completely de novo, but by looking at the fragment ion, we can assign where the core O-glycan linkage sites are, and it's all the way up to 7 glycosylation. And we also done a CID spectrum of the same glycopeptide. CID actually is very complementary to ETD. When we did the CID on the same O-glycopeptide, we saw consecutive neutral loss of the core O-glycans that has a 365 Dalton shift. And then we saw 7 consecutive loss, which indicate that this glycopeptide has 7 modifications coming from the O-glycosylation. So together, CID and ETD gave a lot more information where the glycosylation site and the site heterogeneity. So here's some of the new tools coming out from Waters for glycoprotein characterization. One is standards and reagents. So the standards, the first one we introduced this year is intact monoclonal antibody mass check standards. It's IgG1 protein, and here what the customer or scientist can use is for checking their system performance before they actually do the intact protein analysis. And then here's 2 AB labeled glycan standards. One is the Dextrin letter standard, and the other one is called the glycan performance test standard. So the Dextrin letter and the glycan performance test standard together can be used in the GU retention time assignment. So basically, the scientists would run the 2AB labeled Dextrin letter first and generate a calibration curve can then be applied to their unknown sample. So those 2 glycan standards can be used for system performance purpose, can also be used for GU retention time assignment purpose. And the last I want to touch upon is the informatics tool from Waters. This is a unified biopharmaceutical platform solution. The unified biopharmaceutical platform solution supports LC and MS data workflow in a single solution that encompasses data acquisition, processing, bioinformatics, visualization, reporting, and the configurable compliant tools. So for biopharmaceutical samples that we now in Unify have monoclonal antibody, intact protein, peptide mapping, and the released glycan workflow. Now, the glycan workflow for the released glycan right now, it's focused only on FLR using the Dextran ladder retention time calibration method. But in the next version that will be released this year, that will contain both FLR and a mass spec workflow, and also allowed scientists to use the retention time database, Glycobase 3.1 from NIBR, which will be included in Unified Scientific Library. And here's to show what a glycan FR analytical workflow looks like in Unify. So figure A shows that how a Dextran ladder looks like. We typically use GU4 to GU12 to get the separation calibration curve, shown in figure B. Then in figure C, that just shows how to apply the calibration curve to an unknown sample. Now, once you get the GU value for each peak component for your unknown sample, you can then go outside Unify for now to search in Glycobase 3.0. You can search the GU value, and then it will give you tentative assignment, a list of possibilities. If you combine with exoglycosylase digestions, then you can really get full structure characterization for the glycan. And here's what review panel looks like in Unify after glycan FR data processing. Assuming this is a 3 separate analysis that's done on the same type of sample, the scientists can look at, in this particular plot, is all the peak components being summarized by their relative percent amount within the whole glycan mixture. And you can look at, for example, click on min5 peak, and it will give you also a trending plot, which is very useful. And also you can perform statistical analysis from multiple injections for the same peak component. This is just one of the things you can do in Unify for release glycan. And here's a Unify reporting. This could be very, very useful for any scientist try to communicate their results within their peers in the same laboratory or outside their laboratory. So basically, after the analysis, the scientists can put together a report that has sample list, analytical method summary, the retention time calibration curve from the Dextran ladder, and the chromatographic result table from their unknown sample, and a summary table that combines all injections from release glycan and get statistical analysis. So Unify also in version 1.6, the current version is able to acquire both glycan, UPLC FLR, and the QTOF mass spec data. The scientists can look at accurate mass information from the MS channel. So, for example, this is a 2AB label Intercept M glycans. The top chromatogram is fluorescence, and the bottom is base peak intensity mass spectrometry. I zoom into a particular peak and look at the M over Z value, that this is the G2S1 peak, and has give you the mass confirmation to verify this is the correct assignment. Of course, the fluorescent chromatogram can be subject to the retention time calibration and the GU assignment to further combine with accurate mass measurement. All these will be capable using Unify 1.7 because we will have the Glycobase database 3.1 inside the Unify Scientific Library. Right now, Unify is capable to process FR chromatogram, assign GU value, but you can also acquire the QTOF MS data and look at the accurate mass information. So here's just to show you the product portfolio that we have for biotherapeutic protein glycosylation analysis. We have column chemistry, UPLC H-class, and Xevo G2S for routine LC-MS and optical analysis, and also Synapse G2S HDMS for high-end mass characterization, such as ETD experiment. And then we have informatics solutions, such as Empower, MassLinks, BioPharmaLinks, and Unify System Solution. So in summary, we talked about Waters' solution for glycoprotein characterization, in-type protein glycoprofiling using BioPharmaLinks bioinformatics tool that it allows scientists to do batch data processing, data comparison, for example, batch-to-batch and innovator biosimilar. And also FLR-labeled glycan analysis, the BH glycan column chemistry gives higher resolution and a faster chromatographic separation, and combined exoglycosylated digestion with GU retention time assignment enable full glycan structure characterization. And the UPLC FR Q12MS platform offers glycoprofiling using both FR chromatogram and accurate mass measurement. You can potentially do MS for structure assignment as well. And ETD offers advanced characterization for glycopeptides, and lately we introduced the reference standards that's to help scientists to do system checkup and for getting better LC-MS data quality. And then Unify, we talked about Unify is a platform solution for biopharmaceuticals. And I'd like to acknowledge my colleagues and collaborators from Waters Corporation and NIBRT to work on the glycoprotein solutions and make everything possible. So I would like to thank you for your time and your attention, and back to you, John.
Thank you, Ying-Ching, and thank you, Pauline, for providing so much important and detailed information about glycosylation. Our audience definitely appreciated your talks, and they've sent in some very pertinent questions. Let's get started with our Q&A session.
And actually, before we do get started and take our first question, I'd like to ask the audience to disable your pop-up blockers because a short survey on the webinar will be appearing soon. We would very much appreciate your feedback, as your thoughts will help us to continue to bring you topical and timely webinars in the future. So thank you. So, Pauline, the first question is for you, and the question is, what's your robotic platform for glycan sample prep?
Our robotic platform is a Hamilton Star platform. It's partly because we had one already in the building, also because it's very flexible and allows us to do any kind of glycan release that we would choose to do. There are, I'm sure, other platforms that you could use if you have a more limited need for a particular analysis or something like that. But ours is Hamilton Star.
Okay, Pauline, thank you. And Pauline, where can I get access to the NIBRT glycan unit database?
Yeah, you can simply type glycobase into Google, or you can type in www.glycobase.nibrt.ie. And
Pauline, is there any enzyme for deglycosylation of O-linked oligosaccharides, such as pegnase, as in the case of N-glycans?
Unfortunately, there isn't a generico-glycanase, in spite of the fact that many people have looked, and we ourselves have screened at least 2,500 bacterial cultures looking for one. There is an enzyme which will remove gal-galnac, but you have to have cleaved off any other sugars, monosaccharides, before you use that enzyme. So, in fact, it's not really very useful since you're trimming away the information that you really want to know. So, unfortunately, we're left with having to do a chemical release and the associated degradation of the glycans, which is almost inevitable. Actually, the peeling is mainly from core 1, which helps you to, at least you can see to some extent where the peeled structures have come from. But we would really like someone to discover a generic O-glycanase to match the PNGase F for N-glycans.
Okay. Pauline, and another one, is there an optimal method for calculating GU units? That is, column temperature, mobile phase pH, method gradient. Further, is there a particular column length better suited for making use of GU units for glycan identification?
Yeah, well, you can, the way that you assign glucose units is from the standard ladder. So really you can use any column that you choose. If you're doing preparative work, for example, you might want to use quite a long column. Or if you're doing analytical work, we use, in fact, the 1.7 micrometer BEH columns, which are 21 by 15 millimeters. And so long as you run your standard dextran ladder under the same conditions as you run your experimental sample, the glucose unit values will remain constant, but the temperature and the gradient conditions, if you go onto glycobase, they're all described there. And I think if you haven't got a lot of experience, you maybe just use those conditions because we've chosen those because we've done a lot of optimization. So it's probably better to stay with those temperature conditions and the gradient that we suggest, at least.
And here's a very concise question, Pauline. What flexibility do I have with the GlycoWorks kit?
Oh, that I think is Yin Qing's question. I think she knows more.
Okay, we'll send it over to her. Another question, Pauline. Are there batch-to-batch differences in the glycosylation of monoclonal antibodies that can affect binding specificity and/or KD?
Well, yes. I mean, you would hope that if you were making the same recombinant protein time after time, that you wouldn't get much batch-to-batch variation. But of course, it is really important to check that, because if you do, and you, for example, change the fucosylation level, then you certainly will affect the ability of the KD for binding to Fc gamma R3 on monoclonal antibodies, for example, and you can certainly alter binding specificity if you change the terminal residues, for example, on IgG. If you go from sialic acid, and terminal sialic acid will give a different specificity from IgG that terminates in galactose, for example. So if you have a lot of batch-to-batch variation, it's really going to worry you.
And Pauline, we have one more for you. What do you think about glycan profiling by using lectin columns with different specificities?
Yeah, well, I have to say that personally, it depends. It gives you every technology for looking at glycans gives you different information. The thing about using lectin columns, particularly if you're using mammalian lectins, is that it gives you some idea what in real life what your glycoprotein might bind to. It's often quite confused, because you can imagine that a molecule like erythropoietin has got 3 different sites and a whole range of different glycans at those sites. So it will actually bind to a number of the lectins. So you'll get, depending on the order that you use your lectins and the concentration of the lectin on the column and the concentration of your sugar sample running, your glycoprotein running through, then you will get quite confused data. So on the other hand, if you've only got very limited amounts of material, then doing a lectin array, for example, might give you information that you couldn't really get by any other means if you're very limited in sample. So I think that certainly they've got their uses, and a lot of people have done a lot of good work using them. But in terms of accurate quantitative analytical data, they won't, in my experience anyway, they won't provide that.
Okay, Pauline, thank you very much. Ying-Ching, that very direct question I asked Pauline before, I'm going to ask to you. What flexibility do I have with the GlycoWorks kit?
The GlycoWorks kit, there's two basic formats. One is for high throughput sample processing. So you have a 96-well microelution plate packed with a Helix SPE sorbent, and that's for high throughput. And it also comes with a control intact human IgG protein as a process control. We also have a single-use 1 cc cartridge as a single-use format, not a high-throughput format. Also, that come with intact protein control standard. And so that's the base of the GlycoWorks kit. And then in addition, you can order just the reagent and the chemistry component to supplement the kit. So that would include DTT iodocertamide for protein denaturation, and also labeling chemistry. And also there's accessory that's the standards I talked about, the dextrin ladder and the glycan performance testing, and that's also come in as an accessory. So the customer can choose to just get the basic component that's just a high throughput or single cartridge sample processing, cleanup device, and then along with other accessory enabling chemistry to go with it.
Okay. And we have another question. Does the fluorescent labeling influence the fragmentation of the glycans?
Of course it does, but it's relatively speaking, right? So I assume this person was asking MS fragmentation or collision-induced fragmentation. So 2AB labeled glycan, I think there was also a question about mobile phase. So I typically use 50 millimolar ammonium formate as mobile phase A at pH 4.4. And then the mobile phase B is acetonitrile, and it's running in Helix Helix mode. So what I always use, if I want to collect MS data, I use mass spec compatible mobile phase. So what I generate in positive ion mode is dominant protonated ions. And then when you do subject those ions for CID, you tend to get glycosylated bond cleavage. That's the dominant fragmentation. And then you do get some in a less extent of cross-link cleavage. And that's pretty similar to, say, somebody would run on MALDI and looking at the sodiated ion. So there isn't a huge difference. And, of course, you can do negative ion mode fragmentation as well, and you will still predominantly get glycosylated bond cleavage. I hope I answered that question.
Yes, you did. Thank you. And Ying-Ching, are there batch-to-batch differences in the glycosylation of monoclonal antibodies that can affect binding specificity and or KD?
Maybe Pauline may be a better person to answer that question. I don't know if she's able to answer that right now.
Okay. Actually, there's two questions we have for Pauline, and I think we asked that before, but Pauline, do you want to take that again?
Yes, if that's come through again. Yes, I think I said before, I mean, you hope that you don't get batch-to-batch variations. If you do, you need to look very carefully because the terminal residues on your IgG glycans, for example, can certainly affect affinity for specific receptors like C1q or Fc gamma R3. And if you're looking at erythropoietin, then if you lose sialic acid, then or you have a lot of biantennary glycans, when you're looking for tetraantennary ones, then you can affect the clearance rate. So certainly in IgG and also in recombinant proteins, it's important to keep the glycosylation within quite tight limits.
Okay, and Pauline, somebody has a question. We have a hard time to find the 2AB standard for G1FS1 and G2FS1. Can you recommend a supplier to purchase from?
Yes, actually, I think if you purchase the Waters mix, those two structures are in that mixture. The glycans they're IgG glycans actually. So here we don't really make standard sugars. It's very rare to do that. We usually Sorry, Yingqing.
Yes, I think probably you're correct that those two glycans are included in these glycan performance test standards. It's basically a human IgG glycans spiked in with high mannose 5 and mannose 6. So it should include those two glycans that were asked.
Yeah. So thank you, Yingqing. And just to add that, if you want to make these glycans yourselves, it's not so difficult if you're using HPLC, because obviously it's a preparative method, and you can collect fractions if you have big enough column to load enough material. But very often you don't really need the standard itself. It's enough to have it within a mixture, and I think most of the time we don't isolate individual standards. We just look at their properties within a mixture.
Okay. Well, thank you, Pauline. Unfortunately, we've run out of time, but please note that this webinar will be archived for 6 months on our website, www.genangnews.com. If you missed parts of it, you can watch it again, or you can recommend it to your colleagues and friends. I want to say thanks again to the panel for the outstanding presentations, and I want to say thank you to our audience for your attention and for your very thoughtful questions about various topics brought up during the webinar. And thank you to our sponsor, Waters, who made this event possible.